Hi everyone. I want to thank you for indulging me and allowing me to present this way since my back is acting up and not cooperating with my travel plans. I use this technique with my students and I can now empathize that it's awkward to talk to yourself this way, but I hope it works. I wanted to share with you today some of the research that we're doing on the Ohio River. This research is part of the SENSE grant that you have may heard about in other oral presentations and poster presentations today. This is our NSF EPSCoR grant, Sensing and Educating the Nexus to Sustain Ecosystems in Kentucky and West Virginia. It's a partnership that focuses our resources on the food, water, energy nexus. And this is substantially relevant to the Ohio River system that I'm talking about today, because the Ohio River system has 38 power generating facilities, and about 25% of the watershed is developed for agricultural, pasture, and row crops. It also supports about 160 species of fish, and it provides drinking water to more than 5 million people. So that puts the Ohio River right in the heart of the energy, of the food, energy, water nexus. The Ohio River is about 65% forested. So the agricultural land use being 25% of the watershed represents the largest development or disturbance type of land use in the watershed. There's also 20 locks and dam structures on the river, and 7% of the watershed is developed as developed as urban, industrial, and residential land uses, which you can see here on this map, some of the large cities that are located actually on the river. That's significant to what we're talking about today because we're speaking of factors which might influence productivity, and these towns and cities have municipal waste discharges, which include combined sewer overflow, so they could be contributing significant nutrient loading to the river. So that's something to just keep in mind when we're talking about primary productivity. So the research discussed today, it's going to go towards the overall SENSE objectives of defining the relationship between agricultural production, land use patterns, climate change, energy production, and ecosystem dynamics relative to the water quality in Kentucky and West Virginia's aquatic environment, and also towards leveraging our advanced technologies to better understand and monitor those changes. So specifically today, we're going to be focusing on our efforts on the Greenup Pool of the Ohio River. The Ohio River is just a series of pools, basically, due to the locks and dams. So we're using this pool, which is closest to our university, to be representative of the other pools, and using our infrastructure deployed to evaluate this main objective. Specifically, our attention was brought to this issue. We started down this path in earnest due to a significant algal bloom, which happened in the watershed in 2015. Now, this was a relative surprise because as blooms on rivers, flowing rivers of this size are relatively rare. Sometimes they occur, small blooms will occur localized, but to have one that was 650 miles long and affecting, millions of people, that was an interesting event from troublesome from the drinking water standpoint, but interesting from the research standpoint as to why would this happen on a river of that size. Harmful algal blooms are increasing in lakes and reservoirs, but it just doesn't fit in a flowing river system with what we know of flowing rivers. Our river continuum concept from Van Note would indicate that productivity should be low in a flowing water system, and it fit with Thorpe's river productivity model, but we're not sure about that. We need more research into the shallow areas and what's going on there. The other thing that we're thinking at this time is that how does this river work when it is a series of locks and dams that create this series of pools? Is it functioning more as a lake ecosystem or functioning more as a river ecosystem, and how might that play into the propensity to form harmful algal blooms? So these are the kind of things that we started to consider. Now, when you think about factors that might be influencing harmful algal blooms, immediately you think of nutrient loading. And you can see from this as a small data set that I pulled from the Orsenko website representing 2017, which is the time frame of the data that I'm going to be presenting. And you can see that the nutrient nitrogen and phosphorus are present in the system. They are not limiting in the system, as has been indicated in some of the recent literature that phosphate is not always the limiting nutrient in the ecosystem. Sometimes when you have eutrophication, then the nitrogen fixing bacteria do not keep up as well, and so you can actually get nitrogen as the more limiting factor as well. So the Ohio River system, as I said, is known to be eutrophic, to have high nutrient loading from agricultural areas and the municipalities and things. So we did not believe that this was our limiting factor in productivity going into that, but that is one of the things that is considered. So we begin at the factors influencing productivity by deploying sensors on the upstream and downstream ends of the Greenup Pool, which is our area of study. And you can see here the Robert C. Byrd Lock and Dam is an upstream end, and then the Greenup Hydro Plant and Locks and Dams is on the downstream end. And so this pool here is our study area. We deployed our infrastructure on both of those locations. There's some pictures of our deployment on the Greenup Locks and Dam. And just so you can see here exactly where we are on the structure, at Greenup we are upstream into the Greenup Pool out here on this lock wall. And that's primarily the data that I'm going to be sharing with you today. The sensors that we deployed out there were the YSI multi-parameter exosondes. They measure DO, pH, conductivity, chlorophyll, phycocyanin, and depth. And we had them set to record the data at 15-minute intervals over the the year time frame that I'll be talking about. They're still out there, so we're having more data come in all the time. We also deployed with them data loggers and telemetry so that we could access them from the lab and bring the data back in. And today, when I'm talking to you about some of the metabolism evaluations that we did, we were also using discharge data from the USGS gauging station, which is located in Ironton, Ohio, and we got sunrise sunset data from NOAA. I used an Excel spreadsheet called RiverMat, which in order to do the calculations for the metabolism, primary productivity, and respiration in the Ohio River from our 24-hour DO data set collected with these sondes. And that RiverMet Excel spreadsheet uses the sunrise, sunset, and the discharge data in there. Also, just to mention that I did some modifications to the RiverMet data sheet. I didn't think it was reading all of the sunrise, sunset data and the indexing some of the commands, so I worked on that a little bit, but still use that as the frame. I also want to point out that the year of data that we have, which spans August 2017 to August 2018, that's what I'm talking about today, that was a wet year, and so some of the high precipitation events were not indicative of the average conditions. When I refer to the low flow data, I was using this time frame from early July through the end of August, which was representative of low flow conditions, but frankly, the rest of the year was kind of a wash. Intended. I'm also going to talk about some transect sampling that we did within the Greenup Pool to try to characterize the conditions within the pool. And so on this map, you can see the Greenup Lock and Dam at the downstream location and the bird locks. And we had 22 sampling points scattered throughout the downstream on the river. And those were placed with regard to large tributaries, influences, municipalities, and things like that to characterize the overall conditions on the pool. And what we found from our evaluations right away, we saw that we didn't find much chlorophyll. Our chlorophyll values were quite low throughout all the seasons, and we consider that to be odd. They were, you can see over here, very low. We were expecting 30 plus micrograms per liter of chlorophyll, and we didn't really get in anywhere near that. We did find turbidity as a major factor in the data that we saw, a lot of variation in turbidity. We had, let me just point out on this graph, because I have several that look like this. Across the x-axis, we have the month, and the computer program puts them in order, but our data started here in August, and then so this would be 2017, circling back to 2018, showing an annual cycle. We saw a high variation in temperature, which is exactly what you would expect. You know, it was very warm in July and cold in December and January, so that was expected. And oxygen had an unusual trend that we did not expect. So these data that I'm presenting today are the ones that were used in for comparison with the metabolism data. These are exported from our 15-minute data set, and these are the 2 p.m. measurements that you're looking at here. So this is at 2 p.m. in the afternoon. And in a eutrophic system at 2 p.m. in the afternoon, you would expect to see saturation at least. And here we have oxygen dips in the summer months, which you would expect, but not at 2 p.m. And this is oxygen saturation. And you can see here that even at 2 p.m., you are not supersaturated with oxygen, which you would expect from your algal community. But you are actually showing an oxygen dip, which is unusual. When we look at turbidity, again, as we're considering it month by month, this stands out. And you can see that what the main thing to take away from this graph, well, yes, you do see some higher turbidity in the spring, which you would expect. But what I want to bring your attention to is down here that you always have turbidity. There's not a time when turbidity is low, very low, or reaching zero. So it is a constant presence in this pool. Our monthly chlorophyll concentrations here shown, again, I showed seasonal before, these are monthly. And again, to point out the flat line there, you just don't see peaks overall. You see a few individual days where there were peaks, but by and large, very low chlorophyll concentrations. And when we look at the gross primary productivity and the respiration, you see very similar to what you saw in oxygen concentrations, with the respiration being lower in the higher months, which could be attributed to temperature, and very little gross primary productivity. Peaks, as you would expect during the higher summer months, but relatively flat with gross primary productivity. So what is limiting? What's causing that? The nutrients are present. We just don't see the primary productivity that we're expecting. So we look at two other parameters, flow and turbidity. Now, flow is thought to be limiting in riverine systems for productivity. And so if you look at these graphs, what you can see is the relationship between gross primary productivity and respiration, which are considered to be indicative. A lack of relationship between the two is indicative of flowing water. And as you become more lake-like, that relationship becomes more stronger. And so what this depicts is the overall annual conditions in the river, which are showing no relationship, well, almost, but not a significant relationship between primary productivity and respiration, which indicates that flow could be limiting in the system annually. As you become more, as you go into the summer months, these are the low flow data, July and August, and as you are in these low flow months, you do see this relationship strengthening, which would indicate that the river is becoming more lake-like during that time period, and flow may be exerting less of an influence on the productivity, but it could be a driver in limiting the productivity in the river. I was talking before about the transect data, and this is a representation of our chlorophyll data from the transects that we evaluated in the Greenup pool. And I want to show this to you to talk about the potential for light and turbidity limiting light as a factor in productivity in the green up pool. And so what this graph shows you is that there's no stratification of chlorophyll. The first sample on the top is a surface sample, and then the next line would be 3 meters down, 6 meters down, 12 meters down, etc. This is the bird end, which is the shallow end of the pool, and the green up end, which is the deep end of the pool. And there is no stratification of the chlorophyll concentration at the depths in the pool during this season that we sampled. The turbidity, we also measured photosynthetically active radiation, and there's a significant drop in the PAR from the surface sample to the first sample, which is 3 meters. And so that supports the idea that turbidity and light limitation might be limiting of productivity in the pool. So just further to support that, I love this graph because it shows how messy this kind of data can actually be. But I'm going to bring your attention over here to this end of the graph, which is our low flow conditions from July into August. These are the low flow conditions represented. And I want to show you right here on the graph, which is turbidity. So I've zoomed in on that right here. And what you can see again is what I've indicated, that turbidity never goes to zero. In fact, 90% of the time, the turbidity is greater than 5 NTU. And that 5 NTU has been shown to limit photosynthesis. So this is sufficient to limit photosynthesis even under the low flow conditions. And currently, this is our turbidity is causing this lack of productivity. So we started down this path because we had this harmful algal bloom, and we thought, well, this, overproduction of algae, that is our problem. And as we're now measuring this and looking at this, we're seeing that underproduction of algae is seeming to be a problem. And this is also a problem, as well, because if we're not using the nutrients in the pool and our nutrient spiral is getting longer and longer, then those nutrients are escaping, and they can go on downstream to contribute to problems elsewhere. As you see here, we're located right in here. And so these nutrients can be going on downstream to create problems in the Mississippi, but also these loss of nutrients can create impairment to our fishery, which is, needing algae for productivity. That's the productivity source in our area as well. So we're losing that. So this is a problem. It's kind of right now we have a lot more work to do as we are going forward with our research. We're now trying to get the balance between the loss or the lack of productivity and the potential for the bloom that we've seen in 2015. So we're still collecting data and working additionally on those types of things. So I appreciate your consideration and allowing me to present this way. I have students in the audience. If you have questions, you can turn to them, but they might prefer if you email me rather than put them on the spot. They're there, and please feel free to let me know if you have any questions. Thank you so much for your attention.